At the end of this pre-recorded video lecture, the students are able to learn the different bacterial cell structures and its function. And then we will identify the different bacterial cell shape, size, and your arrangement. But before we go directly to the different bacterial structures and its function, we have to differentiate first what is a prokaryotic cells versus a eukaryotic cells. Remember that bacteria is different from an animal cell, human cell, or even your fungi cell, because as what you can see in this diagram or table, your bacteria belongs to a prokaryotic cell, while your animals, plants, fungi, and protists belongs to your eukaryotic cell. So how can we differentiate your two? How can we differentiate your prokaryotic versus your eukaryotic? First is about your nucleus. Your prokaryotic cells does not contain your nucleus. The nucleus is actually absent, while your eukaryotic cells, the nucleus is present. Now I have a question. Is even though that your prokaryotic cell does not contain your nucleus, does they have a genetic information? Yes or no? Remember, we know that your nucleus does not contain, your prokaryotes does not contain your nucleus. The nucleus is actually absent. My question is, does your prokaryotic cells contain a genetic information? The answer is yes. They still have your genetic information. Even though that your prokaryotics or your prokaryotic cells do not have a nucleus, they do contain your genetic information. Prokaryotes generally have a single circular chromosomes where they store their genetic information. The area that contains this genetic material is what we call as your nucleoid. So dito nakikita natin that even though that they do not have your nucleus, but they have still a genetic information, and this is now what you call your nucleoid. So your prokaryotes have nucleoid, while your eukaryotic cells has nucleus, and it is bounded by a membrane, because your prokaryotic cells, they have no or absent membrane bound organelles, while your eukaryotic cells, they have different organelles. What else po, Doc, ang difference? Another important differentiating factor is that your prokaryotic cells is unicellular, while your eukaryotic cells mostly multicellular, but some are unicellular. So this is actually a misconception, because a lot would think that yes, prokaryotic are really unicellular, but they are thinking or comparing that eukaryotic cells are usually or all the time multicellular. However, there are instances that your eukaryotic cells could also present as your unicellular. So remember, eukaryotic cells can be unicellular. Example of that will be your protozoa, and you will reach your parasitology and you will discuss more of your protozoa. Many people think that your eukaryotes are all multicellular, but this is not the case. While your prokaryotic cells are always unicellular, remember that all of your prokaryotics are unicellular. Eukaryotics can either be multicellular or unicellular. So remember that your prokaryotic cells is always unicellular, while eukaryotic could be multicellular and some are unicellular. Example of that are your protozoans. In terms of cell size, your prokaryotic cells are smaller compared to your eukaryotic cells. In terms of complexity, since it is unicellular and it's much smaller and they have no organelles at all, then they are simpler compared to your eukaryotic cells. In terms of your DNA form, your prokaryotics are circular, while your eukaryotic cells are linear. So again, examples are your bacteria and archaea, and for eukaryotics are your animals, plants, fungi, and your protists. So that is how we differentiate your prokaryotic versus your eukaryotic. Now we understand that the classification of your bacteria belongs to your prokaryotic. So now we will go to the bacterial structure and function. So we will go what are the different structure or different parts of your bacteria, and what are the functions of these different bacterial structure. Tingnan natin. So first, let's talk about these two important terms. We have your essential versus non-essential bacterial structure. So how do we differentiate the two? So example muna ng ating essential, This is your cell wall, membrane, your ribosome, your nucleoid, mesosome, and your periplasm. So you can see that in our diagram. And we have your non-essential. We have your capsules, pilus or pilus, your glycocalyx, your flagellum, your spore, your plasmid, and your granule. So now, Doc, how will you differentiate the two? What is essential versus your non-essential? The term essential actually refers to the structure that the bacterium cannot live without. Therefore, pag sinabi natin essential, kailangan siya for the survival of your bacterium. Without any of these essential structure, then your bacteria will not probably survive. They will not survive because kaya nga tinatawag na essential. On the other hand, pag sinabi nating non-essential components, as the term implies, it is actually an accessory structures. So it is only a nice to have. You may pwedeng wala or pwedeng meron. So a bacterium can possess them or not. However, these different non-essential structures will make your bacterium more pathogenic. They can make your bacteria more infectious. They can make your bacteria survive longer. So later on, malalaman natin why these different non-essential structures makes your bacteria stronger or makes them more pathogenic. So if a bacterium, for example, possesses a non-essential component, it is more virulent. For example, capsule. Your capsule, tingnan natin saan dito ang capsule. So this one, the outer area, your capsule. So your capsule actually inhibits your phagocytosis. Therefore, an infection can easily spread. What else? Your spore or your endospore. Your spore can survive inclement in an environmental condition or in a toxic condition. That's the action of your spore. For example, the bacteria can identify that the environment is not already enough for the growth of a bacteria. Therefore, it will be converted into a spore so that it can live longer, and later on when it observes that it can already grow, then it will transform now from a spore into its vegetative state. So that's how the function of your spore. So again, pag sinabi natin essential, therefore it is essential for the growth of your bacteria. Without it, your bacteria will not survive. Your non-essential is only accessory. The bacterium may possess it or not, but this will help the bacteria becomes more virulent or more pathogenic. So, later on, isa-isahin natin yung mga different structures na ito. So, let's continue. So, first, let's talk about the bacterial cell envelope. So, sa bacterial cell envelope, this covers your cell wall, your plasma membrane, the different layers outside the cell wall. Tingnan natin. Let's talk about first up your cell wall. So, your bacterial cell wall, so the generality, all bacteria have a cell wall composed of your peptidoglycan, except the exception to the rule is your mycoplasma, because your mycoplasma does not have any cell wall, but rather they only have your cell membrane. Your peptidoglycan or your murine is a rigid structure that lies just outside your cell plasma membrane. Mesh-like polymer of identical subunits forming now your long strand. So again, pag na-hear nyo yung salitang peptidoglycan, therefore we can say that it is composed of two important substances. Peptidoglycan, peptide, and glycan. Pag sinabi nating peptide, that's the protein part. Pag sinabi nating glycan, diba? Remember, we have glycan, glycogen, glucose, meaning it is sugar. So therefore, we can say that your peptidoglycan is made of two important substances, the sugar part and the protein part. The sugar part, your glycan, and the peptide or the protein part, which is your peptide chain. So your peptidoglycan, ang kanyang backbone is actually your glycan, plus your peptide side chain cross-linked by your transpeptidases. So, tingnan natin sa ating diagram na to. Your glucose or your glycan is composed of two alternating sugars. So, it is composed of two alternating sugars. Anong dalawang sugar na units na ito? It's your N-acetylglucosamine and your N-acetylmuramic acid. So this is now your two alternating sugars. Tingnan nyo, N-acetylglucosamine alternated with your N-acetylmuramic acid. So tingnan natin, alternating, and also here alternating. So this is now your sugar backbone composed of alternating two units of sugar, your N-acetylglucosamine and N-acetylmuramic acid. What else? And there is also your peptide side chain. So ito yung tinatawag na natin na peptide side chain. This peptide side chain is cross-linked to your sugar molecule by your transpeptidase. Ito yung sinasabi natin na transpeptidase, cross-link siya to your sugar backbone. What else? It is actually composed of your alternating D and L amino acid. So, pag i-compare natin sa 3D configuration na to, titingnan natin that your peptidoglycan is actually in helical shape. So, pag 3D configuration, it is actually in your helical shape. So, tingnan nyo, it's in helical, alternating sugar molecule cross-linked by your different peptide, the alternating delta or D and L amino acid. Very good. So, because of the idea that it is a composed of your sugar molecule plus a cross-link by your peptide chain, therefore, we can say that your cell wall function maintains now the shape of your bacterium. It helps protect the cell from your osmotic lysis and helps protect from your different toxic materials. It may also contribute to the pathogenicity of your bacteria. So that's the basic function. Because of the structure, it basically maintains the shape of your bacterium. So later on, we will observe, we will understand the importance of your gram staining. So we will discuss later on also the idea about gram staining. But right now, I want you to have an idea that your bacteria can be classified whether it is gram positive or gram negative. So later on, i-discuss natin yung gram staining na procedure or somehow the concept or principle behind your gram staining. So one way of classifying our bacteria is actually through your gram staining. Two important results will be your gram positive and gram negative. So your gram staining procedure is actually a staining reaction that will classify them into positive or negative. It will stain actually your cell wall. So now, let's compare the cell wall of a gram-positive bacteria versus the cell wall of your gram-negative bacteria. Tingnan nyo, this orange part, that is actually the cell wall. Just don't mind yet the plasma membrane because we will discuss also your plasma membrane later on, the red one. So focus first on the orange part, this part and this part. So this is your gram-positive. How can you compare your gram-positive now to your gram-negative? Kindly compare it in terms of the presence of your peptidoglycan. Remember that your cell wall is mainly composed of your peptidoglycan. Tingnan nyo ang cell wall ng ating gram-positive. What can you say? What can you see? It is thicker compared to the cell wall of your gram-negative. Diba? Nakikita natin? The cell wall of your gram-positive cell is actually thicker compared to the peptidoglycan or cell wall of your gram-negative. And also, we can see that there is a outer membrane ng ating gram-negative cell na cell wall. I repeat again, pag gram-positive, ang cell wall niya is mainly composed of peptidoglycan, but it is thicker. Tingnan nyo, sobrang kapal. However, the cell wall of your gram-negative cell is composed of your outer membrane. Tingnan nyo, there is an outer membrane and there is a very thin peptidoglycan. So that's the best way for us to compare now that the cell wall of your gram-positive is thicker compared to your gram-negative. Now, dito na papasok of the importance of your gram staining. Again, as always said, that your gram staining is a reaction of your bacterial through their cell wall. Your gram staining is one of identifying factor that we can help to identify a certain bacteria, whether your bacteria, as I said, is a gram-positive or gram-negative. So, paano natin malalaman, or how do we do the procedure? I guess you have a separate discussion with your gram staining. So, I will leave it to the next lecturer, the exact procedure, the different false positive, false negative reaction in your gram staining. But right now, I'll give you an overview, as well as the principle behind your gram staining, so that we can understand or correlate it to the cell wall, since we are talking about your cell wall. So again, pag sinabi natin gram staining, dalawang result, gram positive and gram negative. There are 4 reagents that we are going to use in your gram staining. Remember the mnemonics bias. I know all the medtechs knows about this. So your bias, not bias in your K-pop, but the bias, letter V. V-I-A-S. The word is bias. V stands for violet, which is the crystal violet. So kaya ginawa na lang natin V, no, para mag-make sense ang mnemonic. So V stands for violet, which is the crystal violet. I is iodine. A is alcohol, S is safranin. I repeat, V is for crystal violet, I iodine, A alcohol, S safranin. Now let's talk about their function. Your crystal violet is the primary stain. Your iodine is the mordant. Pag sinabi nating mordant, it is actually it will help the reaction to occur, in terms that in helping your crystal violet stains your bacterial cell wall. Without your mordant, the reaction will not occur. So your iodine will make the reaction occur to make a complex between your stain and your bacterial cell wall. That's the basic definition of a mordant. So iodine is a mordant. A, alcohol is a decolorizer. So later on, we will understand what's the concept between your decolorizer. And your S is your safranin, which is a counter stain. So I repeat, ulit-ulitin natin para ma-memorize na. V, crystal violet, primary stain. I, iodine, which is now your mordant. A, alcohol, decolorizer. And S is your safranin. So basic lang naman ang procedure niyan. You have your smear, prepared smear for a certain bacteria. Then you will apply your crystal violet. You wash that and then put iodine. You wash that, put alcohol. You wash that and put your safranin, and you check that in your microscope. Again, meron tayong 4 na reagent. Crystal violet, iodine, alcohol, and your safranin. So tingnan natin. Your crystal violet will cause now a small dye particle. Tapos, lalagyan mo ng iodine. So, from your crystal violet will react to the cell wall of your bacteria. Then, however, it will only create a small dye particle. Then, you add your iodine, because remember, your iodine will make a bridge to your primary stain to your bacteria. That's why it will now create a large dye complex. After that, you add your alcohol. So your alcohol, if it is a gram positive, it will still maintain a large dye complex. Walang nangyari, walang nagbago. Then you add your counter stain, which is your safranin. When it is a gram positive, it will not cause another color reaction. So therefore, our gram positive will be color purple. Saan nagaling ang kulay na purple na to? Remember your crystal violet. Your crystal violet is a primary stain, so therefore a gram positive will maintain the color of your crystal violet, which is violet. Therefore the result is purple. How about your gram negative? So your gram negative, your crystal violet, again, small dye particle, but with the addition of your iodine, anong nangyayari? It will make the reaction to occur. It will create now a large dye complex. Afterwards, because of the function of your alcohol, what will happen? Since if it is a gram negative, anong mangyayari? Matatanggal yung ating large dye complex. Your large dye complex gets out. So in short, it becomes colorless. Since, diba, purple, naging colorless because of your decolorizer. What will happen? You will now add your secondary stain. Your secondary stain is safranin. What is the color of your safranin? Your safranin is color red. So, anong nangyayari? Since na-decolorize siya, nawala ng kulay, natanggal yung complex natin, you add a color, then the counter stain pale red to red. So, the color is now red to pink. So, that's how we can identify that it is gram-negative. Gram-positive, it maintains the primary color, which is your primary color of your primary stain, which is purple, while your gram-negative, it will now be colorized by your counter stain, which is color red to pink, which is now your safranin. That's why your gram-negative is red. Doc, the question, bakit natatanggal? Why is it that your large dye complex gets out in your gram-negative versus your gram-positive? Again, let's go back to this diagram. Ano nga ang comparison natin sa cell wall ng gram-positive to gram-negative? Remember that the cell wall of your gram-positive is thicker. Makapal ang kanyang peptidoglycan compared to your cell wall of your gram-negative. Sobrang nipis ng kanyang peptidoglycan plus meron siyang outer membrane. So your decolorizer, how does your decolorizer, what will be the effect of your alcohol now? So the gram-positive cell, so once you add your alcohol, first let's talk about dito na part sa ating gram-positive when we add your alcohol. When we add your alcohol to your gram-positive, the gram-positive cell loses some of its large chunky peptidoglycan cell. So, natatanggal din sa gram-positive. However, it can still keep enough of it to retain the purple color. So, hindi lahat natatanggal. Once you add your alcohol sa gram-positive, some of its chunky peptidoglycan cell wall are being sloughed off, natatanggal. Pero, there is still a part of it that is left with a color violet, with a stain of your primary stain, which is now your crystal violet. That's why, even though, Doc, how about, di ba naglalagay na tayo, the step 4, we add safranin as counter stain. So what will happen? When we add your safranin to your gram-positive, it will not actually cause a reaction anymore because we still have your crystal violet attached to your peptidoglycan. The safranin cannot cause any changes in the color because of the presence of your crystal violet in your peptidoglycan na naiwan kanina. How about sa ating gram-negative cell? In our gram-negative cell, what will happen? When we add your alcohol into your gram-negative cell, the outer membrane, this one, ang makapal na part, palakihin natin ha, sorry. Yan. In your gram-negative cell, what will happen? The outer membrane, this part, as well as your peptidoglycan, completely stripped away, leaving it now colorless. So, nagiging colorless. Again, the gram-negative cell has its outer membrane, this one, and your small peptidoglycan layer completely stripped away, leaving it colorless or decolorized. So, nawawala ng kulay, therefore, when we add your counter stain, it becomes now red. So, that is the principle behind your gram staining. I guess you already understand the principle behind your gram staining. It is important for us to identify whether the bacteria is gram-positive or gram-negative. Kindly remember that principle because no matter what, kahit mag-third year na kayo, mag-fourth year, or you're in the hospital, the same principle. Just remember the bias and its effect. And I know you already understand why it becomes purple and why it becomes red for gram-negative. For any clarification or questions in this part, do not hesitate to PM me through my social media account or through your Telegram. So let's continue. So isasahin natin yung cell wall ng ating gram-positive. So the cell wall, let's talk first about the cell wall of your gram-positive. So, composed primarily of your peptidoglycan. So remember, this is the cell wall. So this is the cell membrane of your bacteria. But let's talk first about your cell wall. So this is the cell wall of your gram-positive. Tingnan nyo, it's very thick, right? It's composed primarily of your peptidoglycan. However, it may also contain your teichoic acid. So these are now your teichoic acid. Once your teichoic acid attached to your plasma membrane, it is termed now your lipoteichoic. Remember, your plasma membrane is composed of lipids, kaya tinawag siya na lipoteichoic acid. So, what is the function of your teichoic acid? So, your teichoic acid help maintains your cell envelope, protect from environmental substances, and may bind to your host cell. So, that's the basic function of your teichoic acid. And also, as what you can see, there is a perisplasmic space. It lies between your plasma membrane. So, dito, this is your plasma membrane and your cell wall. It's actually smaller than that of your gram-negative bacteria. So, mas maliit ang periplasmic space ng ating gram-positive compared to the bacteria. However, some textbook would say that there is no periplasmic space to your bacteria. But for the book that I was using, there's still a small, some would say there's still small periplasmic space in your gram-positive. So what is the purpose of that periplasmic space, doc? So it actually contains your enzymes secreted by your gram-positive bacteria are called your exoenzyme, and it will actually aid in your degradation of your large nutrients. Now let's compare the cell wall of your gram-positive now to your gram-negative. So, nakita natin sa diagram kanina that your gram-negative cell wall is more complex compared to your gram-positive. It consists of a thin layer of your peptidoglycan surrounded by an outer membrane. So, thin layer of your peptidoglycan. Where is our peptidoglycan here? This one, di ba? Comparing now to your gram-positive, it's very thin. However, it contains an outer membrane. This part, it contains your outer membrane composed of lipids, lipoproteins, and your lipopolysaccharide. What else? In comparing it to your gram-positive, there is no teichoic acid. So, wala tayong nakitang teichoic or lipoteichoic. So, we see that, remember, diba nasabi natin kanina, it is also composed of your lipopolysaccharide. So, your lipopolysaccharide is actually consists of three parts. Meron siyang lipid A, your core polysaccharide, and your O side chain or your O antigen. So, your lipid A, this part, your lipid A is embedded in the outer membrane, while your core polysaccharide or your O side chain extend out from the cell. So your outer core and your outer or your O antigen extend to the outer part of the cell. So doc, saan po ba dun yung lipopolysaccharide? This is actually now your lipopolysaccharide, parang this kind of filaments. So as you can see, the outer part is composed of your O specific side chain of your polysaccharide. This part that is outside your outer membrane. So let's compare now your gram-positive versus gram-negative based on what we discussed a while ago. For easy comparison, again, the component, peptidoglycan, teichoic acid, and your lipopolysaccharide. Your peptidoglycan to your gram-positive cells is thicker and multi-layer. How about in your gram-negative cells? It is actually thinner and thinner layer. How about your teichoic acid? Your teichoic acid is only present in your gram-positive cells. How about your lipopolysaccharide? Your lipopolysaccharide is only present in your gram-negative cells. So please do not forget this table because this table is very important to differentiate also your gram-positive versus your gram-negative cells. Now let's proceed with your cell membrane. We're done with your cell wall. Here is your cell wall. Now let's go inner. Let's discuss your cell membrane. Your cell membrane, it is a lipid bilayer with floating proteins. So, meron tayong tinatawag na amphipathic lipids which have polar ends and non-polar tails. Pag sinabi nga, when we say polar ends, that's your hydrophilic which interact with water, di ba? Pag hydrophilic, it's water-loving. Pag non-polar tails, that's your hydrophobic which is insoluble in water. So, this is now your cell membrane. This is now your polar ends or your water-loving hydrophilic and your tails which is inside, which is insoluble in water. And also your cell membrane contains now your membranous proteins. What are these different membrane proteins? So we have your peripheral and your integral protein. So saan po dyan, Doc, ang peripheral protein? So this is now your peripheral membrane protein. When we say peripheral membrane protein, it's actually loosely connected to the membrane and can easily be removed. Compared to your integral protein, it is actually embedded within the membrane which can carry out a lot of important function. So that's a basic. Actually, the cell membrane is similar to the cell membrane of a human. So in terms of its function, Doc, it encompasses your cytoplasm. It is also a selective permeable barrier. It also interacts with the different external environment. So meron siyang receptors for detection of and response to chemicals in surrounding, also your transport system and the different metabolic processes. And lastly, in your now let's talk about the different layers outside naman in your cell wall. So first, let's talk about your capsule. So your capsule usually composed of your polysaccharide. It is a well-organized and not easily removed from the cell. Remember your capsule, is it essential or non-essential component of the cell, of your bacterial cell? So please answer, essential or not? It is non-essential. Your cell, your bacterial cell, may survive without the presence of your capsule. Therefore, there are only bacteria that has your capsule. So it is actually visible in your light microscope. So protective advantages, remember that your capsule is non-essential. Therefore, it will make your bacterial cell pathogenic or more virulent because your capsule has protective advantages. It can resist phagocytosis. It can protect from desiccation and can exclude viruses and detergents. So that makes your bacteria more virulent or more pathogenic. We have also your slime layer. Your slime layer is actually similar to your capsule, except that it is diffuse, unorganized, and can easily be removed compared to your capsule. So this diagram, this is actually your Klebsiella pneumoniae with its capsule stained for observation in a light microscope. So there's a special staining method where we can stain your capsule. So nakikita nyo dyan, this is actually the one that is very bright. That's actually now your capsule, the outside, the layers outside your cell wall. Now let's proceed with your Glycocalyx. So again, it is also a layer outside your cell wall. The term that encompasses both your capsules and your slime layers because they usually are composed of polysaccharides. So it's a combination of your capsule and your slime layer. It is synthesized by your surface enzyme. And what is the function of this layer, Doc? It is actually associated with the adhesive properties of your bacterial cell and contains a prominent antigenic site. So, as what you can see in this diagram, tingnan natin. Your bacteria is connected. This is actually your bacteria, another bacteria, another bacteria. And this is actually your intestinal wall. So, this is one is intestinal wall. This is one bacteria, one bacteria, one bacteria, and another bacteria. So, your bacteria is connected to each other to the intestine and also to the intestinal wall. Di ba napapansin natin? This bacteria is connected to this bacteria and this bacteria is also connected to your intestinal wall. They are actually connected by the action of your Glycocalyx, the extensive network of fibers extending from the from the cell. So that is the basic function of your glycocalyx. Adhesive properties of your bacteria. It makes, again, your bacteria virulent, pathogenic. So let's proceed now the bacterial cytoplasmic structure. So we're done. We're ready. Remember, from the outer part, and then we go inside of your bacteria. That's your bacterial cytoplasmic structure. So what are those important structures that I want you to remember? Your ribosomes, your nucleoid, and your plasmids. So remember, your bacterial cytoplasm contains ribosomes and various types of nutritional storage granules. So, however, remember that your bacterial cytoplasm contains no organelles. Remember that. Let's talk about your ribosome. So, similar with human, your ribosome is also complex protein RNA structures. It is also a site of your protein synthesis. However, in comparison to a human or animal ribosome, your bacterial ribosome is actually the complete ribosome is 70 Svedberg, while the complete ribosomes of your eukaryotic is 80 Svedberg. Tinatawag natin the S part of your ribosome is actually Svedberg. So later on we will understand what is Svedberg is all about. Sige. Again, your prokaryotic ribosome, the complete ribosome, tingnan nyo, the complete ribosome is 70 S. And remember, your ribosome could have a small subunit and a large subunit. The small subunit of your prokaryotic cell or your bacteria is actually 30 Svedberg. The large subunit of your prokaryotic bacteria is 50 subunit, but the complete is a total of 70 Svedberg. In comparison to a eukaryotic cell, what is the small subunit in your eukaryotic cell? It's actually 40 Svedberg, while in your large subunit, the eukaryotic large subunit is 60 Svedberg. So remember that, because that is very important to remember. Again, pag prokaryotic or bacterial, small subunit 30, large subunit 50, but the complete subunit is actually 70 Svedberg. For eukaryotic, it is 40 for the small subunit, 60 for the large subunit, and 80 for the complete ribosome. So you just have to add plus 10. Plus 10 ka lang dyan sa result. That's gonna be your eukaryotic. So Doc, what is the importance of that Svedberg? What is the difference of, what is the importance of, what is that Svedberg unit all about? When we say Svedberg, it is actually a non-SI unit. It is actually a unit for your sedimentation rate. So your Svedberg unit comes from your sedimentation rate. Doc, what is sedimentation rate? When we say sedimentation rate, it is actually the rate at which your particles of a given size and shape travel to the bottom of your tube under centrifugal force. Meaning to say, or in simple terms, we have a centrifuge machine. For example, we have a tube with fluid. We put that. Then, because of the action of your centrifugal force, what will happen? The particles coming in your tube will settle down. That's your sedimentation rate. So therefore, your sedimentation rate or your Svedberg will reflect the rate at which a molecule sediment under the centrifugal force of a centrifuge. So meaning to say, it is the rate. We are talking about the rate. So in technical terms, we say that your Svedberg is a measure of time. With that being said, the smaller the Svedberg unit makes it is directly proportional to the speed of your sedimentation. So therefore, it's opposite, not directly proportional. It's actually the opposite. So pag maliit, when the number is lower, therefore it is more faster. Pag malaki, the sedimentation rate is actually lower. So I repeat, pag maliit ang kanyang number, small subunit, mas maliit siya. Remember, it's 30 subunit. Therefore, the sedimentation rate is faster compared now to your large subunit. So that's actually the main explanation of where does this Svedberg unit is actually derived from your sedimentation rate. But that's, I mean, that's very technical on our part, no? But I just want you to remember this one, the ribosome, the importance of the different, the importance that it is for protein synthesis and the different subunits, the 30S and 50S. Why do you have to remember that? Because when you reach in your pharmacology topic, I know that you have a separate discussion on your pharmacology sa ating microbiome, di ba? Part of your syllabus in your microbiology is the discussion of your different antibiotics. There you would understand the importance now of knowing these 30 Svedberg and 50 Svedberg. Why? Because we have different antibiotics that will attack your protein synthesis, specifically your 30S, and attack your protein synthesis, specifically in your 50S. For example, in your 30S, it will be inhibited by your dalawang important drug classification, your aminoglycosides and your tetracyclines. For your 50 subunit, we have your chloramphenicol, your macrolides. Example ng macrolides, we have your erythromycin, your lincosamides, which is your clindamycin, your linezolid, and your cryptogramins. But for now, just I want you to have that kind of idea, the importance of having these different subunits, because the different drug mechanism of your antibiotics will be dependent on these different subunits. Just remember that, of course, hindi mo muna natin alamin yung mga pharmacology topic ng antibiotics. Later on, you will learn about that. So it's a process. Huwag nating madaliin ang lahat. But I guess I have to emphasize the importance of these different units sa ating ribosome. Let's talk about your nucleoid. Again, remember that your prokaryotes does not have contain any nucleus. However, they have this genetic material found in your nucleoid. It is not surrounded by your nuclear membrane. The chromosome consists of a double-stranded, covalently closed circular. This is actually circular DNA molecule. Now, let's talk about your plasmid. So, Doc, what is the importance of this plasmid? So, your plasmid is an extra chromosomal DNA, usually small. So, remember, we have your DNA here in your nucleoid, but again, your plasmid is an extra chromosomal DNA, usually small, closed circular DNA molecules. It exists and replicate independently of your chromosome. It may integrate into chromosome or inherited during cell division. Later on, we will see the importance of your plasmid when we discuss your pilus. Later, makikita natin how this plasmid is transferred from different cells. So this plasmid is actually transferred through your bacterial conjugation and by the help of your sex pilus. So, tingnan natin mamaya. So these are the different major types of your bacterial plasmid. We have your conjugative plasmid, your R plasmid, and your col plasmid. So remember, this is plasmid is a non-essential, meaning it could also make your cell pathogenic. Why, Doc? Because, for example, a conjugative plasmid, it can transfer of DNA from one cell to another. Therefore, it can transfer its specific ability later on to another cell, and that ability may be pathogenic or makes them more pathogenic or more virulent. Then we have your R plasmid. Also, it carry your antibiotic resistance gene. So therefore, your plasmid is responsible actually for your antibiotic resistance. That's the importance of your plasmid. So let's proceed with the different external structures. We have your pili or your fimbriae, and we have your flagella. So your pili, or singular, we have your pilus. It is actually, we have two types. We have your common fimbriae and your sex pili. Your fimbriae or pili is the same lang yan. So when we say your common fimbriae, it's short, thin, hair-like, proteinaceous appendages. It can mediate attachment to the surface, motility, and DNA uptake. So this is now the different fimbriae. Not this one, because this one is actually the flagella. We also discuss that later. That one, very tiny, short, thin, hair-like, proteinaceous appendages. That's your pili. Your sex pili or pilus, it's longer, thicker, and less numerous. The genes for the formation, found on your plasmid, and it is required for conjugation. So, Doc, what is bacterial conjugation? Your conjugation are actually, so your pili is actually involved in your attachment of your donor and recipient bacteria in your gram-negative cell conjugation. So, tingnan natin, what is your bacterial conjugation? This is now your bacterial conjugation. When we say bacterial conjugation, it is the different steps or mechanism of transfer of your plasmid from donor to your recipient cell. So again, pag sinabi natin bacterial conjugation, it is a transfer or it is a mechanism or different processes to transfer your plasmid from one donor to a recipient cell. Remember, your plasmid is one of the most important bacterial non-essential component that's responsible for your antibiotic resistance. So tingnan natin. For example, this donor cell, kaya tinatawag natin F-positive donor cell because it has a specific characteristic that you want to transfer it to another cell. For example, this plasmid contains now your antibiotic resistance ability. Remember, this is an extra chromosomal DNA. So it may be contained. For example, this contains an extra chromosome. It contains the data that it needed for it to become a antibiotic resistant. So antibiotic resistant, for example, this part, your F positive, it has this kind of characteristic, and you want to transfer that to a non-antibiotic resistant na cell. That's why it is F negative. So what are the steps that is needed for that to occur? Number one, your donor cell attaches to a recipient cell with its pilus. The pilus draws the cell together. Remember, this is now the function of your pilus. It will be essential or needed for your bacterial conjugation. Your donor cell will be attached to your recipient cell by the action of your pilus. This is now your conjugation pilus. What will happen next? The cells contact one another. Then, one strand of your plasmid DNA transfers now to the recipient. Tingnan nyo. One strand of your plasmid transfers to the recipient. So, in short, both of these cells contains one strand of your plasmid. Therefore, what will happen next? The recipient, this cell, synthesizes a complementary strand. So, this cell now will be responsible in making now a complementary strand of your plasmid. And then the donor also synthesizes a complementary strand, restoring its complete plasmid. So again, balikan na natin, very basic. Your cell will be attached to this cell by the action of your pilus. Then next, since it will already been attached, it will transfer one strand of your extra chromosomal or your plasmid. And then since one strand na lang, each of the cell will now create the complementary strand. Therefore, the characteristic of your donor cell will now be transferred to your receiver cell, making your receiver cell also resistant. Remember, your plasmid is responsible for making the cell resistant to antibiotics. Therefore, na-transfer niya yung ability niya sa ibang bacterial cell which do not contain those ability. That is the importance of your bacterial conjugation by the action of your pili. I hope you understand that the concept of your bacterial conjugation. Now let's talk about your flagella. Your flagella, it's a thread-like locomotor appendages extending outward from the cell membrane and your cell wall. So the functions we have for motility and swarming, behavior, attachment to your surfaces, and we also, again, remember your flagella is non-essential and may also be a virulence factor. So this is an example of your flagella. This is a monotrichous, meaning one flagella at one end pole of your bacteria. So monotrichous polar flagellation. So your flagella have three important parts. We have your filament, we have your hook, we have your basal body. Your filament, it extends from the cell surface to the tip, followed by a rigid cylinder of your flagellin protein. So saan ang filament? This is now your filament. Your hook, it actually now links your filament to your basal body. Your basal body is actually a series of rings that will drive now your flagellar motor. So that's the basic parts of your flagella. So we have the different arrangement of your flagella. Please also remember this. We have your monotrichus. When we say monotrichus, there is one flagellum. So as what you can see, there is one flagellum at one end pole of your bacteria. We have also your atrichus, meaning they do not contain any flagella. And next we have your Lophotrichus, Amphitricus. When we say Amphitricus, one flagellum at each end of the cell. So tingnan natin, Amphitricus, each end of cell contains now your flagellum. Then we have your Lophotricus. Pag sinabi nating Lophotricus, clusters of flagella at one or both ends. So clusters of flagella at one or both ends. That's your Lophotricus. Actually, your Cephalophricus can also be defined as Lophotricus. So either way, it could be your Lophotricus. So other name of your Cephalophricus, it's still your Lophotricus. Remember, clusters of flagella at one or both ends. So may cluster dito, then may cluster both. So technically, your Cephalophricus, it's still Lophotricus. So don't mind your Cephalophricus. Then we have your Peritricus. It's spread over the entire surface of your cell. So let's have a practice. Kindly identify. What's this? So again, we have already an example of this. This is your Pseudomonas. And we have one end pole of your flagella. One flagella at the end pole. This is now your, is this Monotrichus, Lophotricus, Peritricus, Atricus? This is Monotrichus. Very good. How about this one? We have your flagella, many flagella at one end pole or both end pole. This is now your Lophotricus. And then this is actually your Spirillum. Then we have your flagella all throughout the body of your bacteria. This is Peritricus. Very good. Example of this is your P. vulgaris or your Proteus vulgaris. Now let's proceed with your bacterial endospore. So your endospore, it's a complex dormant structure formed by some bacteria. So there are various locations within your cell. So it is also resistant to numerous environmental conditions. So this will make your cell resistant, pathogenic. So remember, it is resistant to numerous environmental conditions. It can resist the heat, radiation, chemicals, and your desiccation. Your endospores, they have also a location in your bacteria. So depending, pag nasa center part, of course, that will be your central. We have your subterminal, we have your terminal, and we have your swollen sporangium. So what makes it resistant? So what makes it resistant is actually because of its structure. So tingnan natin ang kanyang structure. The spore surrounded by thin covering called your exosporium. So where is your exosporium? This is now your exosporium. Thick layers of protein form the spore coat and cortex. Beneath the coat, thick peptidoglycan. Still, it contains your peptidoglycan. And the core has now your nucleoid and your ribosome. So this part here contains now your nucleoid and your ribosome. Again, what makes it resistant? It has something to do with the component of your structure, because your endospore is composed of calcium complex with your dipicolinic acid, and also it has your DNA binding proteins plus dehydrated core, spore coat, and your exosporium. That is the reason why your endospore is resistant to the different environmental condition. So this is now the process of your sporulation. Pag sinabi natin sporulation, it is a process. Kaya tinawag natin sporulation. It is a process of your endospore formation. So bakit tayo, Doc? Why do the bacteria will undergo a sporulation process? Again, they will undergo a sporulation process normally because when there is instances that the growth ceases because of lack of nutrients, when the bacteria senses that there is not enough nutrient that the bacteria is actually receiving, then it will undergo a sporulation process. Or when the environment is toxic for that bacteria, it will also undergo sporulation. So magiging spore or endospore ang bacteria. So how does that happen? Very easy. Tingnan lang natin. First, your spore. So lakihan na lang natin para makita nyo. So the sporulation process, again, your spore septum begins to isolate newly replicated DNA and a small portion of your cytoplasm. There will be now your spore septum. So meron tayong spore septum na nangyayari. Then what is the next step? Your plasma membrane starts to surround the DNA cytoplasm. So because may contain na siyang plasma membrane in your ribosome there, it will create now your plasma membrane. It will surround now as if it is a new cell. Then what is the next step? There will be now your spore septum surrounds your isolated portion, forming now your spore. Then as time goes by, it will add up the different structures as what we discussed a while ago. Lalagyan ng peptidoglycan, lalagyan ng spore coat until such time that it will be freed from the cell. And that will be now your endospore. So Doc, paano siya after forming now your endospore? What will happen next? Then it will wait. It will wait when the environmental nutrients are being detected. When this endospore can already detect any environmental nutrients, then that's the time that it will now be converted back to its vegetative state. That will now, so babalik na siya sa kanyang pagiging bacteria. So it will now be converted to its vegetative state. Your spore now will rupture. So meron na naman yung process na pagdadaanan. It will undergo activation, germination, and lastly, growth. That's the vegetative counterpart. So again, once there is no enough nutrients coming from the bacteria, therefore, if the bacteria will not have enough nutrients, therefore the bacteria will undergo sporulation. And then after such time, when your endospore is being freed from the cell and already detect nutrients from the environment, therefore it will be converted to its vegetative state. It will now be freed again. The spore will rupture. It will undergo different stages of your vegetation, magiging activated, germinated, and it will grow again as a bacterial cell. That's the basic mechanism of your sporulation. Again, not all bacteria will undergo sporulation. There are only few bacteria that will undergo sporulation. Example, we have your Bacillus anthracis. We have also some species of your Clostridium. So remember the two important, your Bacillus and your Clostridium. They will undergo sporulation. Again, this will not be essential for the bacteria, but rather these are non-essential components. Therefore, it will make again your bacteria more virulent, more pathogenic, and or making them more resistant to other different environmental factors. Okay, we're done with the different bacterial structure and its function. I guess that's a very basic concept that I want you to understand, so that when you read your book, it's easier for you to grasp the concept. So let's have a review. So tingnan natin. Please try to answer, because this is the only way for you or for yourself to assess that somehow you really did understand my discussion a while ago. So try to answer, ha? It contained the genes that confer protective properties such as antibiotic resistance, virulence factors, or their own transmissibility to other bacteria. Again, contains the gene that confer protective properties such as, ito talaga, antibiotic resistance. Paulit-ulit na lang to. This is now your very good plasmid. So please try to answer. Just try it. Hindi ko na naman maririnig mga sagot nyo. So just try it in your home, in your study area. At least you will really assess yourself. Number 2, involved in protein biosynthesis and are membrane bound. So protein biosynthesis. This is now your very good ribosome. Next, selective permeability and active transport facilitated by membrane-bound permeases, binding proteins, and various transport system. This is now your, similar to the humans, this is now your cell membrane or plasma membrane. Number 4, it is unique to prokaryotes. It is found in all bacterial cell wall except mycoplasma. This is now your peptidoglycan. So your cell wall, remember your cell wall is composed of your peptidoglycan. Protects the bacteria from phagocytosis and plays a role in your bacterial adherence. So again, protects from phagocytosis. This is now your, very good, your capsule. And lastly, form as a survival response to a certain adverse nutritional conditions. This is now your endospore. So very good. That is a very important concept or things that you really need to learn. Now, let's proceed with our last discussion or last topic. Konti na lang to. So, this is now parang the laboratory part, the lab part. Kasi parang lecture part yung ginawa natin kanina. But this is really now the laboratory part. So, now let's talk about the size, the shape, and the arrangement of your bacteria. So, your size, shape, and arrangement, pag sinabi nating sizes of your bacteria still varies even though that they are very small, but their sizes really varies. The shape, there are two most common shapes. We have your cocci and your rods, but there are still various others shape. Then arrangement, pag sinabi nating arrangement, this is in comparison to their in comparison to the other bacteria. So paano sila nagkatabi-tabi? So this is actually determined by your plane of division and determined by separation or not. Tingnan natin. So, let's talk about the sizes. So, again, the sizes, the smallest is your mycoplasma. The average rod is approximately this size, and the very large, okay, just don't mind that. So, in comparison to your red blood cell, your E. coli, which is an average rod, is smaller than your red blood cell. So, remember, your RBC is approximately 7,000 nanometer in diameter in comparison to your E. coli, which is 1,300 by 4,000 in nanometer. So, mas maliit siya compared to your red blood cells. And also, mas maliit ang yung Streptococcus and mas maliit pa talaga ang ating mga viruses. So, the different shape and arrangement. So, what are the different bacterial shape? We are talking about your cocci, your bacilli, rod shape, and your spiral. Then, each of these bacterial shape will have its own corresponding arrangement. So, pag ang iba-ibang arrangement ng ating cocci, we have your coccus, we have your diplococci, tetrad, sarcina, streptococci, staphylococci. The arrangement of your bacilli, we have your bacillus, diplobacilli, streptobacilli, coccobacilli, your pallisids, and for the arrangement of your spiral, we have your vibrio, spirochetes, spirilla, which is helical shape, corkscrew form. So, tingnan natin isa-isa. So, let's talk about first your cocci. When we say cocci, it is sphere. Coccus present as single cell. The first arrangement is coccus present as single cell. So, meaning pa isa-isa. That's your coccus. Again, the singular of your cocci is coccus. Pag sinabi natin coccus as an arrangement, present as a single cell, pa isa-isa. So, this group includes bacteria that are present as a single cell. So, pa isa-isa. Ulit ka naman, Doc, eh. So, tingnan natin. After your coccus, we have your diplococci. When we say diplococci, it's actually in pairs. So, this arrangement results when two bacterial cells occurs as a pair joined together. So, two bacterial joined together. Tingnan nyo. As what you can see, lakihan ko ha, para mas ma-view natin. Yan. Your bacterial cells occurs as pair. Tingnan nyo, as what you can see, it occurs as pair, right? Buti pang diplococci may pair. Sorry. Tingnan nyo, it is two bacterial cells occurs as a pair joined together. So, diplococci. That's your diplococci. Joined together. So, what are the example of this? You have your Streptococcus pneumoniae, your Moraxella, your Enterococcus, and your Neisseria gonorrhea. So, don't mind your example. I want you to really first understand the different shape and arrangement. So, tingnan natin ha, your different joined together. So, is this gram positive or gram negative? Sige na, let's apply what we previously learned. This is actually a gram stain of your sputum. So this one are your bacteria. Remember, these are cocci. So therefore, this is gram-positive or gram-negative. This is very good. Remember, it maintains the purple color. This is now your gram-positive. So pag nilagay natin sa result, ang ilalagay natin, gram-positive diplococci or gram-positive gram-positive cocci in pairs. So either way, pwede nang ganyan pagkasulat. So gram-positive diplococci or gram-positive in pairs. Very good. Now we have also your chains. So this is the arrangement now. Your chains, or your example of that, your streptococci. So here, the bacteria are actually arranged in long chains. So, excuse me. So this actually belongs to the present family of your of your Streptococcaceae, so which is characterized by a lack of motility and a gram-positive bacteria. So tingnan natin, very basic, long chains. So this is now your Streptococci arranged in long chains. So again, is this gram-positive or gram-negative? Of course, this is now your gram-positive. So gram-positive cocci in chains or gram-positive streptococci. So this diagram, this is actually your Streptococcus agalactiaceae, the one that is responsible for your group B streptococcal infection. This is a cocci arranged in chains, color enhancing, using now your color enhancing electron micrograph. So using your EM microscope. I never had the opportunity to see or view in your electron microscope. Never tried. Ang ganda sana very, very graphic. Next, we have now your cocci in grape-like clusters. So you have your staphylococci. So this results from cell division in both planes that are characterized by organisms which are immotile and gram-positive. So again, kaya nga tinawag na clusters because they are grouped in clusters. So parang grapes. So as what you can see, these are in grapes. Is this gram-positive or gram-negative? Still, this is purple. Therefore, this is gram-positive. Again, what is the reason why it becomes why it is in clusters or in groups? Because this is actually the result of your cell division in both the planes that are in both the planes. Next, we have your tetrads. We have your micrococci. So these are 4 cocci in square. So very basic. These are tetrad bacteria that are arranged in a group of 4 cells that remains attached and grow in the attachment after your cell division. So this arrangement results when the cells divide in your 2 different planes. So very simple lang. 4 cocci in square. This is now your tetrads or your micrococci. Next, we have your sarcinae or your sarcina. This is a cubic configuration of 8 cocci. So again, pag tetrad or apat, that's your tetrads. Pag walo, cubic configuration of 8 cocci, as what you can see here, this is now your sarcinae. So the bacterial cells forms a group of 8 cells. So bakit nangyayari to, Doc? Because this happens when your cells divide in a perpendicular plane. That's why in a cubic configuration, makikita natin na it is actually composed of your 8 cocci. So let's review again. So review pairs or your diplococci, chains or your streptococci, your grape-like structures which is your staphylococci, your tetrads which is your micrococci, 4 cocci in square, or your sarcinae which is a cubic configuration of your 8 cocci. So let's have a practice. So let's try, let's identify. I want you to practice yourself because again, this is the only way for you to really identify if you really understand my lesson. So what's this? Single lang ang ating mga cells, ang ating mga cocci. First, identify what is the shape. This is cocci or bacilli? Of course, this is cocci. So this is cocci. Now the question is, what is the arrangement? What is the arrangement of this one? This is a coccus as individual cell. This one, is this gram-negative or gram-positive? This is, of course, naka-red siya. This is your gram-negative. this is gram-negative. This is now your, remember, in pairs, joined together, two cocci joined together or in pairs, this is now your diplococci. So, pag sinabi natin in a result form, gram-negative cocci in pairs or gram-negative diplococci. Very good. This one, gram-positive or gram-negative? Again, this is gram-negative. Gram-negative cocci in chains. This is now your streptococci. Very good. Gram-negative streptococci. Very good. This one, this is actually in 3D configuration. This is composed of 8 cocci joined together. This is now your sarcina. Very good. Gram-positive sarcina. How about this one? In clusters, first identify gram-negative, gram-positive. This is your gram-negative. So, gram-negative cocci, this is now your staphylococci. But guys, remember always remember that the cocci could either be gram-positive or gram-negative. So, depending on the reaction. But in generality, pag we say staphylococcus or staphylococci, it's actually gram-positive, not this one. This is actually incorrect. Remember that, ha? Sige lang. You will learn the different characteristics. You will learn the different characteristics when you go along the discussion in your microbiology. But for now, I just want you to learn the basic concept in terms of the sizes and the shape. How about this one? These are actually your tetrads. 4 combinations of your 4 groups or 4 cocci in a group. Okay, very good. Now let's proceed with your bacilli. So pag sinabi nating bacilli kanina, spheres. Pag sinabi nating bacilli, it's actually in rod shape. So bacillus are rod shape and are present as single cell. So pag sinabi nating bacillus, again, single cell. So these bacteria can form endospores and are facultative anaerobe. So example of these are your salmonella, your bacillus, and different salmonella such as salmonella cholerae, salmonella enterica. So don't mind the different examples, but first I just want you to learn really the shape and arrangement first. So the bacillus, single cells. In comparison sa ating cocci, yung coccus natin. So as single cells. We have your diplobacilli. Remember, we have your diplococci. Again, pag sinabi nating diplobacilli, it's actually rods in pairs. So, the bacilli that exist in pairs. Is this gram-positive or gram-negative? This is actually gram-negative bacilli. I'll give you a generality in terms of your gram staining. Your gram staining, pag sinabi nating gram-positive, usually they are cocci. Pag gram-positive, generally cocci. Pag gram-negative, generally your bacilli. However, there will always be an exception to the rule. Excuse me. Not all cocci are gram-positive. There are exceptions to the rule. And also, not all bacilli are gram-negative, but majority. So, in general, pag gram-positive, remember first of your cocci. Pag gram-negative, remember first of your bacilli. But this is not always the case because there will always be an exception to the rule. You will learn that. Next, we have your coccobacilli. Your coccobacilli is a very short rods. This is in comparison, ha? This is your bacillus. This is now your rods. This is your coccus. But your coccobacilli are very short rods. Mas maliit sa bacillus, but greater than your coccus. As the name suggests, coccobacilli, it will now resemble both your cocci and your bacilli. So again, streptobacilli. So similar with your streptococci. Pag strepto, what does that mean? Arranged in chain. So tingnan natin. Lakihan ko lang ang diagram para mas makita nyo. Remember, this is question. Gram-positive or gram-negative? Of course, red siya. This is gram-negative. Then identify the shape. Is this bacilli or cocci? Of course, this is bacilli. Now identify the arrangement. This is actually in chain. So therefore, the complete answer for this one is that it is gram-negative bacilli in chains or gram-negative streptobacilli. Very good. Now we have your palisades, which resemble now your picket fence structure. So this is now your palisade. So these are type of bacilli that resemble a picket fence structure as a result of a bent at the point of division. So because of a bent during your cell division, it will create a it will now resemble as your picket fence. So they appear similar to your Chinese letter. So parang sa gram staining, ito ang nakikita natin. Actually, this is not a gram stain. This is a special stain for your Corynebacterium. Pag sinabi natin mga Chinese letters, always, usually, talagang tinatandaan ko that is actually a Corynebacterium. That is very unique for your Corynebacterium. That's how the arrangement of your Corynebacterium, Chinese letters. So your palisade. Remember that. So let's practice. So identify what is this shape. This is now your picket fence. Bago lang natin diniscuss, or Chinese letter. Example of that is your Corynebacterium. We have your palisades. This one, shorter than your rods but bigger than your coccus, resemble both of your rods and your cocci. This is now your Coccobacillus. This one, bacilli in chains. This is now your Streptobacilli. Bacilli in pairs. This is now your Diplobacilli. Singular for your bacilli, this is now your bacillus. Very good. Now for our last arrangement, we have your spiral. So first we talk about your Vibrio. So your Vibrio is a curved bacteria. So remember, once a curved bacteria, it's a comma-shaped Vibrio. So this group includes the bacteria that are either helical shape or curved shape or comma shape. The bacteria can range slightly curved to a corkscrew like your spiral. So these are slightly curved bacteria resembling now your comma shape. Example of these are your different Vibrios. Your Vibrio parahemolyticus, Vibrio cholera. So remember, pag Vibrio, it's a curved bacteria. Your spirochetes, these are spiral bacterial having a helical na shape. Pag helical shape, this is now your spirochete. So these are, sorry, nalobat pa lang laptop ko, sorry. These are flexible and have an eggshell which helps in motility. So these filaments are actually essential in distinguishing character between your spirochetes and your other bacteria. So these filaments run through the length of your bacteria, thus help in twisting the motion of your bacteria. So example of your spirochetes, remember your TBL, your Treponema, your Borrelia, and your Leptospira. So remember that three that belongs to your spirochetes. They have a spiral bacteria having a helical shape. Tatlo yan ha, TBL, your Treponema, your Borrelia, and your Leptospira. And we have your spirilla, which is also similar in structure with your spirochetes, but they are more rigid. They too have a flagellum, but they lack the endoflagella in your spirochete. So example of that, of your spiral shape, is your Campylobacter jejuni. So again, let's practice. Again, check natin lahat-lahat. Identify. This one is your coccus. Pag in pairs, this is now your diplococci. This one are your tetrads. This one is your pag configuration or 3D pag walong. This is now your sarcina. Clusters, sorry. Naguluhan ako sinasabi ko. Pag in clusters, this is actually your staphylococci. Pag in chains, this is now your streptococci. How about the arrangement of your bacilli? A single one, we have your bacillus. Pag in pairs na bacillus, diplobacillus. Then we have your streptobacilli. Then your palisades and your coccobacilli. How about for the arrangement of your spiral? We have now your, first, this is now your spirochetes. This is now your spirilla. And lastly, this is now your vibrio. Very good. We still have other shapes and arrangement. So we have others. So we have also your mga filamentous bacteria, as what you can see in your gram stain, parang really a filament. So these are bacteria that are long, thin, and filament shaped. They sometimes divide to form branches resembling strands of your hair or your spaghetti, called your mycelium. So example of this is your actinomycetes. We have also your mga pleomorphic bacteria. When we say pleomorphic bacteria, they do not have a specified shape that are included in this group. Walang specified na shape. So we have a lot of example of that. A very good example of that is still your mycoplasma. So again, last, for your quiz, expect this kind of quiz, ha? Remember, I want you to really learn. So expect this kind of quiz. First, identify the gram staining reaction, then the shape, and lastly, the arrangement. We have the first one, this one. Identify. This is gram. Sige, lakihan ko, ha. Excuse me, sorry. Order. Oh. Yan. So this one, your gram-positive cocci, this is gram-positive or gram-negative? Of course, this is gram-positive. Is it bacilli or cocci? Of course, very obvious. This is sphere. So this is cocci. So how about the arrangement? The arrangement is actually in chains. So this is a gram-positive cocci in chains or gram-positive streptococci. Very good. Number 2. This one. Is this gram-positive or gram-negative? Again, this is gram-positive. Is this how about is this a cocci or bacilli? Of course, this is a cocci. What is the arrangement? Of course, as what you can see, they are actually in pairs. Tingnan nyo, dalawang cocci magkatabi, dalawang cocci magkatabi. So therefore, this is a gram-positive diplococci or gram-positive cocci in pairs. Very good. Number 3. Again, is this gram-negative or gram-positive? Of course, this is gram-negative. Is this cocci or bacilli? This is cocci. Tapos, in pair. So this is gram-negative diplococci or gram-negative cocci in pair. So again, diba, yung generate na sinasabi ko, not all gram-positive are cocci because some of your cocci are gram-negative. This is actually your example for that. Last. Very obvious. Gram-positive or gram-negative? Of course, this is gram-positive. The arrangement, the shape first. This is bacilli. Then, the arrangement in chain. So, therefore, this is gram-positive bacilli in chains. Very good. I guess you already understand the basic concept about how to check for the shape and arrangement of your bacteria. We already able to identify the different bacterial structures and its function. So that will actually end our discussion for today. So I want you to still read your book because still the book has the complete details of these topics. So kindly read your book. So if you have any questions for my discussion today, do not hesitate to PM me. If you're still confused on certain part, please do not hesitate to PM me through my social media account. So you can PM me to my messenger or through my Telegram account. So I hope you learned something from today. Thank you everyone and God bless you. Again, for my quiz, again, you have to really identify, you have to learn the more of the laboratory part na tayo pagdating sa ating quiz. So, thank you everyone. God bless you.